Hello everyone. Today we'll be looking at a problem involving meiosis, and specifically how we go through meiosis when an error occurs during it. Let's take a look at problem number 1 here. Below are the cells of an organism undergoing meiotic division, so of course we know they're going through meiosis, and they're currently in metaphase 2. So metaphase 2 means they've already divided once, and now the chromosomes are aligning along the metaphase plate to prepare for the second division. However, during anaphase 1, so when the chromosomes were initially being separated to opposite poles of the cell for the first division, a nondisjunction error occurred, and the homologous chromosomes were split incorrectly. Now, what is a nondisjunction error? A nondisjunction error is when homologous chromosomes or sister chromatids, and in our case, it's homologous chromosomes, fail to segregate during anaphase, and so both homologous chromosomes are pulled to one side of the cell and so are found only in one of the two daughter cells. And in this case, we can see that with our two green chromosomes right there. So our first question is asking, how many chromosomes does each cell have? And this is pretty straightforward. Each of these are one chromosome, and even though you might be tempted to count them as two because they are replicated and contain two sister chromatids, they're each one chromosome. So cell number 1 here has 1, 2, 3, 4, 5 chromosomes, and we will write that down. And cell number 2 here has 1, 2, 3 chromosomes. Next, we're asked how many tetrads are here. Well, a tetrad is a pair of homologous chromosomes that we would see in metaphase 1 preparing for the first division. However, we're in metaphase 2, so we won't see any tetrads here. And even though we see a pair of homologous chromosomes in our nondisjunction error chromosomes over there, they are not paired together for division, so we would not consider them tetrads. So here, both cells have 0 tetrads. And next we're asked how many centromeres are here. Well, that's as simple as there being 1 centromere per chromosome. So in cell number 1, we have 5 centromeres, and we only have 3 in cell number 2. And again, this uneven distribution is because of our nondisjunction error that occurred during the first division. And finally, how many chromatids do we have here? Well, as I mentioned earlier, each of these chromosomes is composed of 2 chromatids. So cell number 1 has 5 chromosomes and therefore 10 chromatids. And cell number 2 has 3 chromosomes and therefore 6 chromatids. All right, now let's take a look at problem number 2. How are the chromosomes assorted before the first division in metaphase 1, and where did the nondisjunction error occur? Well, we know in metaphase 1 that homologous chromosomes align themselves on the metaphase plate at the center of the cell. And looking at our question number 1, we can see that there are 1, 2, 3, 4 different kinds of chromosomes here. We have the blue, red, green, and pink chromosome. So all we have to do is draw them in pairs along our cell. Once we've filled out the chromosomes like this, we can answer the rest of the questions. So where did the nondisjunction error occur? Well, we saw back in question 1 that the 2 green chromosomes were split incorrectly and not segregated during the first division from each other. And so we know that those 2 green chromosomes is where the error occurred. And next we're asked in part B, how many chromosomes, chromatids, centromeres, and tetrads were present? So in terms of chromosomes, again, we can just count here. We have 1, 2, 3, 4, 5, 6, 7, 8. So we have 8 chromosomes. Next, we're asked how many chromatids we have here. And again, that's simply twice the number of chromosomes, as these are replicated chromosomes. They each consist of 2 sister chromatids. So we have 16 chromatids. And next, centromeres. Each of these chromosomes possesses 1 centromere, so we will have 8 centromeres. And how many tetrads are present? Well, here we do actually have tetrads. We can see that these homologous chromosomes are paired together, forming a tetrad, and we have 1, 2, 3, 4 tetrads. And finally, we're asked, what is the diploid number of this organism? Well, we know a diploid organism has 2 N chromosomes, where N is the number of different chromosomes. So we see 1, 2, 3, 4 kinds of chromosomes here. 2 times 4 gives us 8 as our diploid number for this organism. All right, let's move on to our final question, question number 3. So here we're asked to draw the 4 gametes produced as a result of the cells in question 1. So if we go look at question 1, we can take a look at these 2 cells, and we can basically split up the chromosomes that we see in each of these cells into their sister chromatids, which will become the chromosomes for the 4 gametes. So each of these cells will produce 2 gametes. So if we look at cell number 1 here, we see it has a blue, red, 2 green, and a pink chromosome. And cell number 2 has a blue, red, and pink chromosome. So let's split those into their chromatids, which will become the chromosomes for our 4 gametes, and we'll draw those. So now that we've drawn our 4 gametes, let's answer the rest of these questions. How many tetrads are here? Well, there's no tetrads. Tetrads, again, are pairs of homologous chromosomes, and they would have 4 chromatids each. Here, there are only individual chromosomes. Chromatids as well, we have none. So chromatids are part of 2 halves of a chromosome, and now that they've separated, they're no longer chromatids, they're entirely chromosomes unto themselves. And how many chromosomes are present in each of these gametes? Well, again, we just have to count here. So 1, 2, 3, 4, 5 chromosomes in gamete number 1, and the same for gamete number 2. But we can see, because of that nondisjunction error, that gametes 3 and 4 are both lacking a chromosome, and they only have 3 chromosomes each. We're then asked, what is the ploidy of these gametes? So we know that gametes produced by meiosis are haploid. So if our diploid cell was 2n equals 8, then our haploid cells will be n equals 4. However, in this case, we see that there's some differences because of the nondisjunction error. So gametes 1 and 2 have 5 chromosomes, whereas gametes 3 and 4 only have 3. So in this case, gametes 1 and 2 are n plus 1 for their ploidy, and gametes 3 and 4 are n minus 1, as they have 1 less chromosome than what we would expect. And that kind of answers the final question here. How many chromosomes would be in each gamete had meiosis occurred without error? Well, because our haploid number is n equals 4, there would be 4 chromosomes in each gamete. All right, that's all for this video. Thank you for watching.